Dear students, today I am going to take class on Cryptosporidium. As I have already told you that whenever you uh, go to read any parasite, you must have some vague idea about the classification of the parasite. And as you know, the classification is quite cumbersome and it is quite intricate. So you must have some vague idea about the classification. So I have told you that there are four phylums in when it comes to protozoa. Those four phylums are Sarcomastigophora, Epicomplexa, Microspora, Celiophora. So the parasite which I am going to teach you, it comes into the phylum Epicomplexa. And the epicomplexa, when you go for the vague classification of epicomplexa, it can be divided into three major portions. One portion is blood epicomplexa, in which Thaleria, Babesia like parasites come. The second one is systemic epicomplexa, in which you have read the Toxoplasma gondii, Neospora caninum, and Sarcocystis like parasites, and intestinal epicomplexa as well, in which I have taught you already media species. So Cryptosporidium, which I am going to teach you right now, it comes into the intestinal epicomplexa as well. It is a Cryptosporidium is now regarded as a very important emerging zoonotic parasite. Actually, uh, the cases uh, the cases are increased uh, every year. And as far as the history is concerned, the Cryptosporidium was first recognized by Tizer in 1907 from a mice, and he named that species as Cryptosporidium muris. In 1912, the Tizer, recognized the second species called the Cryptosporidium parvum from the mice itself. Since then, there are several species. I remember there are 25 species has been reported on the basis of forced specificity, but International Commission of Geological Nomenclature recognized only 14 species. Several species are of zoonotic importance. There are several genotypes of the species, which I am not going to discuss here. You just remember that there are 14 established species of Cryptosporidium, and the as I have told you that it is an emerging zoonotic parasite. So the first case of first human case was reported in 1976 in United States of America, when 3 years old child in USA has been suffering from diarrhea continuously for 2 weeks. And the public health implication of this parasite has caught attention in 1993, when there was a major waterborne outbreak took place in the United States of America, in the Milwaukee in the state of Milwaukee, which is in, I think it is in Wisconsin. It was the first major outbreak due to Cryptosporidium. And that particular in that particular outbreak, 4 lakhs people were 4 lakhs people were infected, in which 4,500 people have been hospitalized. There were 100 deaths reported, and the estimated loss was actually more than 65 million $5 million. So this was the first major outbreak due to Cryptosporidium. And after that, this parasite has got attention, and the several research took place regarding the importance of this parasite. So when investigated, it was found that the oocyst of the Cryptosporidium, which was actually very small, somehow crossed the filter of the several water filtration plants. So that finding have forced the many countries to to change their water filtration system, and there are millions and millions of dollars are spent to install the filter which can remove the oocyst of cryptosporidium from the contaminated water. So one thing is important here, that one thing which I would like to tell you, that the oocyst of cryptosporidium is actually one of the smallest oocyst when it comes to several parasites of coccidia. So after that, cryptosporidium has got the uh, national attention, and it has been regarded as the potential cause of many waterborne diseases. Our several drinking water reservoirs such as pond, river, well can be contaminated by infected animal feces. This parasite has a wide host range as well. So, untreated water from the river used for drinking water can be act as a source of infection for us if it is. And there are several water sources like swimming pools and water parks have also been incriminated as a source of infection. As I have already told you, that size is very small, and one more important thing is that its oocyst is very robust oocyst, and it is very resistant to extremes of the temperature, and extremes of temperature like it can survive up to 0 degree centigrade, and in the upper range 65 degrees centigrade of temperature it can resist. It resistant to chlorination. It cannot be destroyed. Even the 10% formalin solution and 5% ammonium solution is required to kill the oocyst of this parasite. If you use the concentration less than this recommended concentration, you cannot kill the oocyst of cryptosporidium parasite. As I have already told you that there are several, uh, I think 24 recognized species of cryptosporidium. So there are several species like cryptosporidium suis, cryptosporidium phallis, cryptosporidium canis, cryptosporidium miliagridis in birds. But there are three very important species of cryptosporidium, which are zoonotic importance as well. That is the Cryptosporidium bovis, that is responsible for cryptosporiditis in bovine, Cryptosporidium parvum. Cryptosporidium parvum is actually regarded as a highly zoonotic parasites, and found in many cases of cryptosporidiums in humans, in calf, actually in sheep and goat as well. The third one is Cryptosporidium andersoni. Formerly, it was known as Cryptosporidium muris. It can also infect the cattle, the ruminants, and even the mice as well. So Cryptosporidium has a very wide host range, and the several species of this parasites lacks host specificity, so that cross contamination, or you can say that cross infection easily takes place between the animals, when the ruminants, animals, lab animals, and human. Actually, this parasite uh, mainly parasitizes the microvillus brush border of the gastrointestinal epithelium of wide range of vertebrates, including humans. And this parasite is actually responsible for I tell you, responsible for severe gastrointestinal disturbances, hemorrhages, and mucoid diarrhea, anorexia, loss of condition, malnutrition, malabsorption, etc. So it is quite important one. Now we will discuss the life cycle of Cryptosporidium.